Bon, donc merci à vous tous de vous être joints à ce séminaire virtuel. Je suis très contente que Dr. Diana Baralle a accepté de se joindre à nous sous ce format. La présentation sera en anglais. On demande à tout le monde de fermer vos caméras et vos micros pour le temps de la conférence. Aussi, à la fin, si vous avez des questions, nous les envoyer par chat. Et moi et Christine Van de Velde, on va transmettre les questions pour Diana. Donc, je vais maintenant ensuite changer en anglais pour introduire notre conférencière. So, Dr. Diana Baralle holds a prestigious NIHR research professorship in genomic medicine, a personal chair at the University of Southampton, as well as being a consultant in clinical genetics, working at the interface of research and clinical service. During her undergraduate training in medicine at University College London, she completed a degree in human genetics at the Home of Genetics, the Galton Laboratory. She trained first in pediatrics and then clinical genetics. She completed her doctorate and Action Medical Research Training Fellowship in the Department of Pathology at Cambridge University, where she held an academic position before her current appointment in Southampton. Professor Baralle runs a translational research group that drives forward functional genomics to improve diagnosis. She investigates novel causes of rare disorders, and in particular, the role of RNA and splicing in genetic disease. Thank you so much, Dr. Baralle, to join us. It's a pleasure to have you virtually in Montreal and looking forward to your talk.
So, thank you very much for having me. I wish I was in Montreal. Thank you. So thank you very much for having me. So I'm a clinical academic, as per the introduction. So I sort of have legs in both camps, and hopefully I'm going to share with you the story of trying to how to work towards making new and better diagnoses. So the challenge hopefully is not new to any of us. We've been trying to address this for many years. We want to find sequence variants that are mutations. And if you look here at all the variants listed in ClinVar, you can see that 48% are listed as of uncertain significance, so sort of unclassified. And that means that we can't do anything with them clinically. So that's a large proportion. So I want to say thank you to Leah Starita from the States that gave me this particular slide. And it is a year out of date, but it still has the same message. That basically we're much better at reading DNA than understanding what those changes mean that we find. And nowhere is this more true than for missense variants in genes. So for a variant of unknown significance to be deemed pathogenic, we need to know its effect. So here on this chart, on the y-axis, is the total number of missense variants we find, and the bubble size, you can see getting bigger and bigger. That's more and more missense variants in particular genes, and red is cancer, green are cardiac genes, and blue is other. Probably what I work in most, which is rare diseases. So you can see that actually as the number of genetic tests increase, we keep finding more and more missense variants. And the bubbles just keep getting bigger, they never get smaller. So if we pause the animation there and we label some of these genes up, you can see that actually they're genes that we all know very well. They've been, they're famous genes, they've been well studied, and yet we still don't understand what variants in them mean. So you can see it here, it stops at 2019 and not 2020. And all these variants that we can't interpret represent missed opportunities where we could either have provided needed care or assure a family that actually they're not at risk and they don't, for example, need to have a mastectomy. And this causes confusion in the clinic. So a recent study showed that 50% of surgeons would have offered surgery to women with a variant of unknown significance in a BRCA gene, even when the ACMG guidelines say that you should not change management according to a variant of unknown significance. So this is really important. So why are we missing mutations? Well, I'm sure there are many reasons. I think a good proportion of them we're missing because we do not take into account splicing, the role of splicing from transcription to translation. We're sort of missing out that step when we look at DNA. Because at the moment, we're just thinking of sequence with a huge emphasis on its role in protein coding only. And sequence is needed for other reasons too. It has regulatory regions. It has sequence that splice factors have to attach to. And of course, we are still not interpreting the non-coding region. We call it non-coding, but actually there's important things in there too. So if we look at splicing, so this is the process of removing introns and ligating exons together to make the message that will be translated into the protein. There are some basic splicing points. So at there's the 5' splice site, the 3' splice site, and there are particular splice factors that attach to that sequence, the U1 at the 5', the U2 complex at the 3'. But there's also splice regulatory elements deep within exons. There are enhancer elements within exons. There are silencer elements and various factors that bind there. And these also exist within introns. So if, for example, you have a mutation that falls into a regulatory element that should be an enhancer and you disrupt that enhancer, then that will be a mutation, even if it's a synonymous change. And splicing is complex. It makes you dizzy. There are so many factors involved. And it's not just the factors bind into the sequence, but the structure that it then creates is important. The speed that it's read at is important. And it's much more, this should be in 3D, and I don't have a 3D picture of this, but there's a lot going on, and we do not understand all of it. So I'm going to use an example from my own work, but there are plenty out there. It's just convenient to use my own. So this is a lady I saw with a disorder called neurofibromatosis type 1. And when we sequenced her gene, she has a variant deep within the intron, so nearly 300 bases into the intron. Is this the cause of her NF1 or not? So I put it into a mini gene to be able to see what was being transcribed. And as you can see from the gel, I hope, there's a funny band compared to the wild type. And when we did a lot of work on this, I did a lot of work on this, but essentially there's an area of the intron where the variant falls into that it didn't take much to turn it into an exon. So in fact, it was a highly conserved area with a very strong splice site, and now a region of that intron is able to bind a splice factor called PTB and make it into an exon. So this was now intronic exonization and causing an abnormal protein and the disease. So that's my own example, but here I've listed some papers with examples in cancer and in muscular dystrophy. And in all these papers, you will see that synonymous, missense, and intronic changes can cause disease. And yet in a lot of our large-scale, high-throughput, next-generation sequencing, when it comes to clinical analysis, our bioinformatics will filter these out and we will miss these mutations. So can we look at RNA in a high throughput manner? And can we do this for a health service so that we are able to increase our diagnostic rate? So there's many factors to think about and take into account. How do we do this? So ideally it should be high throughput, generating mini genes is not high throughput, but for example, an RNA-seq analysis might be a good way of developing an RNA diagnostic pipeline to look at these VUSs. And I think that if we do that, we'll be able to interpret and classify these variants of unknown significance. I also think that we can develop RNA sequencing to find mutations where we don't have an easy VUS candidate, that actually it will point us in the right direction. It's possible that by RNA-seq, we can have signatures for a disease or signatures that treatment has worked. I'll show you later that by doing this, you may find new genes associated with disease and possibly new avenues for genetic treatment. I won't go into it because I haven't got the time, but for example, the Spinraza SMA splice therapy is the first that's been extremely successful in humans, but this really lends itself to sort of one-off personalized medicine therapy. So I'm going to tell you about the study that we undertook to try and prove that we really need this in a diagnostic situation. And I thought that if we did this systematically, prospectively, and looked at many cases, that I could build up enough evidence for people to change their practice. So we collected over 250 clinically suspicious variants of unknown significance from UK clinical genetics physicians. And I didn't give them any criteria for sending them to me, only that they thought that the gene that the VUS was in would fit with that patient's phenotype. I didn't want them to do any bioinformatics. It didn't matter whether it was in the coding or non-coding region, just sort of the right ballpark of the gene. And I wanted to do three things. So we undertook an RT-PCR, the cheap, easy way to look at splicing. We wanted to see the same result on RNA sequencing to see if it was able to pick it up, find the same thing, and if it was easy enough to do in a clinical scenario. And we wanted to look at the bioinformatics for splicing, because up until now it hadn't been very helpful in a clinical situation. The variants were scattered across exons and some in introns, and the picture here shows where they fell. So only 27%, less than a third, were actually in a spliced region. And the blue bar shows the position and the number of variants that we had at that position. And the orange bar shows the number that affected splicing. And we collected the RNA in patch gene tubes, again, to try and make it simple, to stabilize the RNA, and you can post patch gene tubes even if you were, you know, far away in Cornwall or somewhere like that. And this has been published in June this year in Genetics and Medicine, if you wanted to go and have a look. But what we found was that actually a lot of them did affect splicing. So we had a third that caused aberrant splicings. That's 85 of the 257 variants. Most of them caused an upstream exon skipping, so 39 of that 85. But we also had some that caused other things, and I'll give you some stats on that now. So of the variants that were in the splice donor region, the splice site donor region, 77% of them caused aberrant splicing. And those that fell into the splice acceptor region, 68% of them caused aberrant splicing. Only 15, so when I said to you before, the most common thing was upstream exon skipping, and that was 39 of them, 15 of them caused downstream exon usage. And there was one, there are some a little more complicated. There's one double upstream exon skipping. That sometimes happens when exons are regulated together and are very close to each other. And we had 4 that were multiple, more complex events. So I thought I'd talk first about the bioinformatics splicing prediction. So how well do our clinically used splice prediction tools perform? So most UK labs, I don't know in Canada, but I believe it's the same, Alamet is used. Now this is not an open access, easy to use system, but it incorporates various tools, including MaxEntScan, NN-Splice, and SSF. And because they don't perform very well, usually labs want a consensus from these tools. So if 2 out of 3 tools say the same thing, then you might take it seriously, or 3 out of 5 tools, for example. So it's really heavy work and not very accurate. So if we look at, I mean, we now had a great resource. We had 250 variants that we had shown functionally caused, whether or not they caused aberrant splicing. So we can compare that with what the bioinformatics was saying. And they were much of a muchness. So if you look at this rock plot here, assessing the accuracy of the predictions, the ideal line goes straight to the top in the graph and along the top. Okay, that would be the ideal tool. The grey dotted line is random chance. And you can see they all have pretty similar accuracy. But we also assessed a machine learning tool called Splice AI, developed by Illumina. And this was published last January. And as you can see in the pink dots, it was much better. And in fact, the area under the curve, the accuracy is 91% compared to in the, between 80 and 85. So this tool alone was much better. So what is Splice AI? Well, it's a machine learning tool, and the diagram shows what happens when it's training. And it's trained on primary transcripts from GenCode. And the nucleotide sequence is fed into this convolutional neural network, and I'm not a computer science person, so I've had to learn to understand it. And it makes predictions on whether the splice sites are there or not, and compare it to what it's seen in its training model and whether they're true or not. And it's tweaked and run again and again until it's as good as it can be. And if I show you, so the paper is there below, but if I show you some of the results they were seeing in these initial papers, they have deployed it in the Genomics England 100,000 Genomes project, initially on non-intellectual disability epilepsy cases only, so that was 12,000 cases. And they give themselves different stringency scores. So, for example, they have their own score of Splice AI 0.2 or 0.5. So 0.5 is the more stringent level. And AF cutoff means how rare that variant is in EXAC or NOMAD. So again, 0.001 is the more stringent. So if we look at the more stringent criteria here, what we would have expected, was to see 117 splice sites, and we actually see 258, so there is a discrepancy there. So these would be sort of high confidence candidates that we need to then do some RNA sequencing on and validate. And they're continuing this, and the plan is that we will go on and prove whether the RNA sequencing matches their calls. But I'm much more optimistic about this tool than any of the tools that have gone before. So much so that I have a PhD student exploring this AI methodology at the moment and how we can improve it. So for example, Splice AI only uses the primary transcript, and Ron, my PhD student, is trying to use a collapse model with all the transcripts and all the exons, and so far he can improve the tool. And the example here is a gene called SMAD11. So as well as trying to improve it, we're trying to see what's happening in this machine learning black box, what's it seeing, and also to analyze its errors. So what's happening with the other 9% that it's not picking up. And hopefully these approaches will be hypothesis generating and maybe teach us something about the biology of splicing. So I thought I would give you some examples of what we found. So here, this gene DKC1 causes dyskeratosis congenita, and you can see we have a variant at +10. We wouldn't be able to tell whether that was pathogenic or not, but in fact, it's using alternative splice sites. So one of the interesting things about this case was that we could not see this on RT-PCR because the change was too small on the gel. So this was picked up on RNA-seq. So actually here we show that RNA-seq picks up things that RT-PCR couldn't. Back to my favorite gene, neurofibromatosis type 1, and here we've got a +3 change. Some of these do cause aberrant splicing, some of them don't, so you really do have to look. And here I think it's probably because it's disrupting the binding of U1, and you can see that quite strong shift to the abnormal transcript in the patient in the gel just down here. So this example in a gene that causes osteogenesis imperfecta, the P3H1. Here we have a really complex scenario with intron retention, alternative 5' splice site, and then another transcript with an alternative 5' and 3' splice site. The control band is quite weak here, but you can see it does have those two bands, the normal splicing and an intron retention band. But in our patient, we've got two other bands here in the middle. And my final example is SF3B4, which causes this craniofacial problem. And here, this is a synonymous variant of unknown significance and actually quite deep within exon 3. So it's 254 nucleotides into the exon. And this caused the creation of alternative deep exon donor sites. So a new donor site well into the exon. But then because the splicing process wanted to use that one, it then decided to use another site deep within the exon as the acceptor site. So here we've got a sort of an intra-exon novel exon. So we've called it an excitron, but it is different from the control. And in fact, this has also been studied by Minigene. So it's definitely there. Oh, and some more intra-exon variants here within the BRCA2 and the BRCA1 gene. So well into the exon and causing exon skipping. So they must be affecting some sort of enhancer or silencer. So, well, can we just set up RNA sequencing in a diagnostic lab? So there's lots of research and decisions really that need to be made on the way. Things like, should we do long read, short read sequencing? Should we do total RNA? Should we just do mRNA? How deep? How long? What else? Once you've decided on the sequencing and all the experimental design of that, what's the bioinformatic pipeline design? That's really important as well. And of course, all this has to be put together with the biological knowledge of the gene and the phenotypic knowledge from the doctor, because all together you make the decision. It's not, right, let's sequence this gene, press the button and it gives you the answer. I'm afraid it's not that easy. So we've got a few projects going at the moment to try and answer some of these questions. So in this one, we're working with the Enigma Consortium, and this is a consortium of people that try and look at variants in cancer genes. So here, 8 BRCA1 and BRCA2 known mutation samples were sent blinded to 11 labs around the world. And I'm one of them. And we have to find what the mutation is using RNA sequencing without knowing what it is and using our own design of the experiment. And then we're going to see if everybody finds the same. So that will be fun. For example, here we have a +5 change, and you can see here on the sashimi plot, the exon skip down below here in the blue. And what we found so far is that we haven't got all the results back yet, but the study has shown that so far RNA sequencing gives us a really good picture of the expressed transcripts. and in a quantitative way. But there is some variation in splicing results between different protocols and different platforms. So here is principal component analysis of all the lab's results, and it's not neat and tidy at all. We have, with the Genomics England 100,000 Genomes project, we have an RNA GSIP, and GSIP stands for Genome Clinical Interpretation Partnership, and I lead that. And we want to use the 100,000 genome resource to also show the utility of transcriptomics analysis. They don't have 100,000 RNA samples, but they do have about 25,000 RNA samples, and they have ethics to recontact cases. So we're working with them and Illumina to select a pilot. First 5,000, Illumina are paying, and we're trying to enrich this group with some positive controls and definitely cases that have been negative on genome where people can't find the result. So we'll see how that goes. But I wanted to also say that doing this work and being a clinician means that I've been lucky enough to find new genes associated with human disease as a byproduct of this work, really. So here, last year in clinic, I saw a consanguineous Romani family, 2 sisters with severe developmental delay, small head and cataracts. And I asked for a clinical exome, and that came back negative. But when you look through the data, and of course you have to have time and you have to have the knowledge to look through the data, you could see that there was a homozygous variant of significance in a gene called COPB1 that had never been reported in human disease. So I looked at the family tree. So there were 2 sisters affected, and then in a consanguineous relationship, there were another 2 cousins that had died with similar signs. And then I looked at the gene, and it seems to be a very important gene that's involved in the protein transport between the ER and the Golgi. So it looked pretty good. And in the literature, there were other cotopathies. That's other genes causing, COP-B genes causing disease. So there was COP-B2 that caused microcephaly, so that was very interesting, and COP-A that caused autoimmune arthritis. So again, there seemed to be a good background of this. And why I was particularly interested, because this homozygous gene change was a +1. Now, I have had +1s before, so at the splice site, that didn't cause disease. So we still have to prove it. And we did a quickish RT-PCR, and it did cause exon skipping. But this still isn't enough to prove this is the cause, particularly when it's not been published before. So I have some lovely colleagues close by who run the European Xenopus facility, and he said that he would knock this gene out in his tadpoles. And it was little more than a week. And when he knocked the gene out, COPB1, in his tadpoles, he gave me tadpoles with small heads and cataracts, which was astonishing. He said he's very rarely seen cataracts in frogs. Of course, they were consanguineous families, so I was sitting there thinking maybe the cataracts are caused by one gene and the small head and intellectual disability caused by another. But here we show that actually they're part of the same syndrome. That's still not enough to say that this is the cause, really, because you have to go out there looking for other families. You need a series of them. And we managed to identify other families by looking through the DDD project and through GeneMatcher. And now we are very confident that this is the cause for this family. And as an aside, I just want to say that this is really a genomics revolution for us, because here's another one. These 3 patients at the top panel were mine in clinic, and I paid for an exome. A frame shift mutation came back in a gene that had never been reported in humans before. And I collaborated again with the Xenopus facility and found people in France that worked on this gene anyway, because they're interested in it in terms of neurons. And together we could show that actually this gene caused disease too. And one last one, if you will be patient with me, because this was an early one from 2014. And I sent this off for an exome. Again, a variant came back in a gene called Pura, never before reported. I searched around for other cases, found some more, 3 or 4 other cases, and then had a fellow that wanted to write it up. But at the same time, we heard that another centre in the States wanted to, had a group as well. So then you either join up, which they weren't very keen on, or you just race to write. And myself and this fellow wrote this paper in a weekend, submitted it on the 30th of September, and it was accepted with revisions by the 8th of October. So I think that's my quickest paper ever. It was quite a hard weekend. Oh, and I love this gene as well because I've got to know the families very well, and they've been extremely helpful in raising funds, in getting families together. It's been a pleasure. And then you never know what genes do, but it looks like this gene is actually a spliceosome protein as it interacts with SRSF1. So that's now generating a whole new branch of research. So back to splicing. Mine isn't the only evidence out there. There's plenty of other evidence. There's evidence in your own institution with Martin's work. But I will show you 3 quite important papers here. There's this one from 2017 where they found new diagnoses by transcriptome sequencing of muscle tissue, so neuromuscular disorders. And I like this one, Frassard et al., because they didn't keep to a category of disease. They just did a bunch of cases they didn't have a diagnosis in with a transcriptome. And they got an 8.5% pickup of diagnoses. And interestingly, even in disorders where blood isn't the main tissue. So it does show that we can do this in blood, which is obviously the easiest tissue to get hold of. Oh, and this one was during lockdown. So this is from Saudi Arabia, again, picking up a whole bunch of new cases in genome biology. So what can we do now? Well, we have a problem. Everybody asks about leaky splicing, what level is pathogenic, and this is really difficult to build into guidelines. I suspect it's different for different genes and different exons, so we need to work that out. It's not only for, my second point is that it's not only just for making diagnoses, but also for maybe reclassifying variants that we've classified wrongly in the past. And I was involved in this paper in 2016, where this -2 variant in BRCA1 was originally thought to be pathogenic. And actually, we had quite a lot of evidence from our mini gene studies in our lab that it wasn't changing splicing. And in this case, the transcript that was being made was still functional and actually rescuing the change. So actually, this did shake the diagnostic world a little bit, because they had to go back to women and say this wasn't the cause of their familial breast cancer. And also, I haven't shown you, but I can tell you that I have plenty of examples of canonical splice site mutations that are not always pathogenic. So just be careful there. So how do we fit this into any sort of clinical guideline for people? Well, it's been incredibly difficult. We spent quite a lot of time last year toing and froing trying to do this. And there is some sort of guideline actually now published in this paper in the Journal of Medical Genetics. It is part of this CanVig group, multidisciplinary group, looking at variants in cancer. And our guidelines are buried deep in the supplementary material if you want to have a look. There was a lot of arguing about this. But basically, PS3 is the functional side of the ACMG guidelines, how you can use functional work to either move your variant up into pathogenic or down into benign. So the summary is that if you have two orthogonal assays, independent assays, showing abnormal transcripts with no evidence of leakiness, that's very strong evidence, and you can, move your variant up into pathogenic. Strong evidence is one assay with abnormal transcripts and no evidence of leakiness. And moderate evidence is one assay or more with abnormal transcripts and evidence of some leakiness. Now, I would say that nearly all my assays fall into that, into moderate, and that it's actually going to be very hard with clinical criteria to move any variant into the sort of pathogenic strong, very strong. And even with those criteria, this box here on the left are all the, yes, but if this happens, then you should do this. And this isn't, what is the level of leakiness, 80-20, 20-80. So I'm not going to read them all out, but I can tell you we spent hours in meetings trying to decide what these assays should be, and ended up with 12 points for people to read through, which is not very user-friendly. And I suspect that all these guidelines will have to change. They'll need adapting pretty soon, I should think, because this doesn't talk about things like RNA sequencing, and we're learning really rapidly how this works. So just to put it in big print there for you to have a look at, I think that it can often be supporting evidence or moderate evidence. So I think it's going to be really hard to put it up into the red boxes there. So I'm close to the end, but I just wanted to talk to you about coronavirus, because I'm a rare disease physician. That's what I see in clinic. But during our coronavirus lockdown between March and June, we didn't see many patients, so I saw them on a screen. I wasn't able to examine them. And our wet lab, I don't know if yours did, but our wet lab shut down for 3 months. We weren't allowed to do any experiments. But we did have quite a lot of RNA-seq data. And we had a project where we were looking at mutations in primary ciliary dyskinesia genes using nasal brushings as the tissue. These nasal brushings are in an air liquid culture, an alley culture. And my PhD student had been looking at gene expressions, PCD gene expressions during time, what time of the alley culture was it best to look at the genes to find mutations. So he had quite a lot of data. So we looked at how SARS-CoV-2 was thought to work, and we went back to our data because we had data from the airways. So I've just got 2 slides, and it is very premature data. But what we found was there was a novel isoform of ACE2 in the human nose and in the bronchial respiratory epithelia that was upregulated in response to RNA respiratory virus infection. So what did we know? We know that angiotensin converting enzyme 2 is the main entry point in airways for SARS, for COVID-19. ACE2 binds the protein spike and triggers the viral fusion with the cell membrane, resulting in the viral RNA genome delivery into the host. And up until now, ACE2 was thought to encode 5 transcripts, but we could see another one, a novel short isoform in the nose and in the airways. And this isoform was substantially upregulated in response to interferon stimulation, which of course is one of the treatments at the moment that is working for COVID, and respiratory virus infection, but not in response to SARS-CoV-2. So here you can see it on the sashimi. And actually, if you look at the gene itself, the short isoform here lacks the spike glycoprotein high affinity binding site. So we think this is consistent with the model where the short ACE2 isoform can influence the host susceptibility to SARS-CoV-2, and potentially there's some therapeutic strategies where this can be manipulated, for example. So even though I'm not an immunology person at all, or infectious disease person at all, we entertained ourselves in COVID lockdown using our own data and learnt new skills. So in summary, really, we're missing mutations. We need to find them. And I think we're missing them because we're not thinking about what that sequence does. It's not just for protein coding. We're not thinking about RNA. And if we did that, we would uplift our diagnostic rate. We would be able to reclassify variants of unknown significance and help patients. We'll be able to identify mutations in this non-coding space. And, there's this big drive to do genomes, but we need to, use it. I think the machine learning is going to really change our bioinformatics. And it's been a surprise to me how many new disease genes we are finding by doing this. And I suspect we'll be able to open new therapeutic avenues. So I want to say thank you to my funder, the NIHR, and Genomics England, our labs, Illumina, the European Xenopus facility and the University of Southampton. This is our team, and I always look at it and think how short I am compared to everybody else, but they're a great team. Thank you very much.
Thank you so much, Diana, for your great talk. Does anyone has question for Diana? If you have, please write it in the chat. So I might start with one. So I wanted to know, like, of all the gene where you had found alternative splicing mutations, have you looked at expression? Like, does it decrease the expression of the genes to the transcript go through an MD, or it causes mostly like a small or shorter isoform?
Mostly it causes isoform, but it might be because of the way we look at it. So in the next iteration, we will do different bioinformatics on the RNA-seq to look at expression, in more detail. I've just seen the next question, Sarah. Yes, of course. I've been working with so her question is, would long read sequencing be better? And I agree. And I think somebody needs to do it, systematically for clinical use. At the moment, people have been doing it in pieces. And I've been really working with what I had at my fingertips, which was the Illumina, because they were paying for the sequencing. So that's what I had with me. But I would love to get some long read done. I have another COVID project happening, which was 100 COVID RNAs compared to 100 flu RNA-seqs. And in that project, we're doing short read and long read together. So we'll be able to see what the difference is. And already there, we can see some, some genes popping up in the COVID that aren't popping up in the flu. So that'll come. Yes, so do you envisage that the RNA-based genetic diagnosis can be done alone without a parallel DNA? I really hope so. I think for some genes that will really be helpful. That's a really difficult question. How do you propose to rule out the leak effect? It's really difficult. We have to have more evidence, more experience, and then we might be able to make the call better. But yes, we need more experimentation.
Are there any other questions for Diana? So maybe a more general question. So for exome sequencing in the clinics, there's some really good guideline, and usually clinical lab likes that things are pretty much the same and can be repeated from patients to patients. But the analysis of splicing is a lot more complex, and there's still not one protocol that can be applied to all disease type or tissue type. So how do you see the future of RNA in clinical lab, and how clinical lab will be able to adapt to this more complex type of analysis?
Yeah. So I think we need to spend the next little while working out how best to do it. So before this talk, I had many questions with the students. So one of them was, you know, about the depth, the length, and the analysis, and we just don't know quite how to get that right. But we have done some work on what the sort of minimum depth is to see 80% of the splice junctions. So then you know you really have to go that deep. But there might be some, we're going to have to get to know our genes better, because for some of them we might have to go deeper. I've been concentrating on blood because I know that that's easy for doctors to get hold of. So if we could generate, with evidence, the best protocol, standard operating procedure for blood RNA-seq for a majority of genes, and I know it won't be ideal, but I think if we could capture 80 to 90%, then that's better than we're doing now. And I think all of us need to work hard to answer all those questions on that seesaw. You know, what should the protocol be? And you could do it for muscle. Because, you know, we need to do it for other tissues too.
Yeah. Thank you. Anyone have other questions for Diana? I don't see any questions, so
I understand why, because we had an hour of questions before.
yes, exactly. So thank you so much, Diana, for participating in our seminar. It was a pleasure to have you with us virtually, and it was a great talk. So thank you so much.
And thank you for having me.
Bye.
Bye-bye.